Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to talk about Hekton enteric medium. Okay, so Hekton enteric medium is again the type of the culture media which we specially used for the isolation of mostly gram-negative rods. Okay, so it's specially used for the isolation of gram-negative rods. So we can say it is not a selective media, means it is actually a selective media for gram negative, because on this media we cannot grow the gram positive bacteria. Okay, so this is actually, we can also say that it is the differential media as well, because on this media we can grow only and only gram negative enteric bacteria, gram negative rod shaped bacteria. So this media was first of all proposed by a scientist named King and Metzger at Hekton Institute. That's why the name is Hekton enteric medium. Then let's talk about the uses of this media. We specially use this media for the isolation of Salmonella, Shigella, that are mostly the gastrointestinal pathogens, and we can also isolate this type of the bacteria from food, water, as well as from fecal sample using this media. So it means that Salmonella and Shigella, which is actually the two types of the bacteria, so we can also say that this media is also a selective media for this, but it doesn't mean that you will isolate the Salmonella and Shigella. You can also use this type of media for the other gram negative rods as well. Now let's talk about the composition of this media. This media contain protease, peptone, ammonical, iron citrate, sodium chloride, distilled water, lactose, we have fuchsin acid, agar, bromothymol blue, sucrose, we have salicine, bile salt, and yeast extract. Now these are the most important ingredient, and these are the amount of ingredient which we need for 1 liter. Now let's talk about the principle of this media, means what are the most important function of the ingredient that are present in this media. First of all, we have protease peptone. The protease peptone is actually act as a nitrogen source, means it can provide the nitrogen source in the media that is specially need for the fastidious bacteria, means the growth of the fastidious bacteria. Then we have the carbohydrate like lactose, sucrose, and salicine. So whenever we using these types of the carbohydrate, it is specially used for the identification of coliform bacteria. Then we have bromothymol blue. This bromothymol blue is actually nothing but it's a kind of a color which we add, and this is actually the indicator. And remember guys, it is red in color. So when the bacteria that produce acid, so it change the color of the media from red to yellow. Okay, and it is also possible by the presence of aldehyde. Then we have yeast extract that can provide what? That can provide energy by which the bacteria can grow and divided. Then we have sodium thiosulfate and iron citrate. This sodium thiosulfate and iron citrate mostly produce the hydrogen sulfide production. It is the black colonies of the bacteria which is appear on this media. It is possible by the iron sulfide. Okay. Then we have what? We have the presence of bile salt and some dyes. So the presence of bile salt and dyes mostly important for the gram positive bacterial inhibition, and thus we have gram negative bacilli to develop on this media. And finally we have agar that is used as a solidifying agent. Let's talk that how we can prepare this media. Now to prepare this media we have to use 72.66 gram of the agar, means we need the powder, and we have to mix it into 1 liter distilled water. Then after that we have to heat mix and dissolve it instead of autoclaving, because we not autoclave this media. If we autoclave, so all the ingredient will be disturbed. And after the temperature, after the heat mix and dissolve, so then we have to cool it at 50 degrees Celsius. After reaching to 50 degrees Celsius, what happened? We have to pour it into petri plate. Now as I already talked that it is specially used for the Salmonella and Shigella bacteria isolation. But we can also say that other gram-negative rods like E. coli, enterobacter and Klebsiella, Pseudomonas, Aeromonas hydrophila bacteria can be also grow on this media, except gram-positive bacteria. So gram-positive bacteria cannot be grown in this media due to the presence of what we can say dyes and bisalt. Now let's see that how the gram-negative E. coli will be look like. E. coli mostly produce large size yellow to salmon color colonies. Enterobacteria and Klebsiella mostly produce large mucoid size yellow to salmon colony. Salmonella produce green color colonies with black center. Green colony with black center. Shigella produce green colonies. Pseudomonas produce irregular green to brown colonies. Aeromonas produce yellow colonies because it ferment the sucrose. And obviously the gram-positive bacteria not grow. Now let's see the diagram of each bacteria which we talked. So these are the growth of Salmonella, as you can see, Shigella, sorry, these are the growth of Shigella. As you can see, these are the green colonies. Now these are the yellowish colony due to the fermentation of the sucrose. These are the colonies of Aeromonas hydrophila. Then we have enteric bacteria, enterobacter and Klebsiella. So mostly both are same, but Klebsiella usually produce mucoid colony as compared to enterobacter. Then we have Pseudomonas, as you can see, irregular colonies of the Pseudomonas. And we have what? We have the E. coli. So these are the salmon color of the E. coli, orange like color. And we have the Salmonella, which can produce green colonies with black center. So that's all about the Hekton enteric agar medium. I hope you guys get idea about that. Thank you so much for watching.